Good afternoon. Is coming for one workshop and symposium कम workshop AI के बारे में कुछ सिखाएगा वहीं पे workshop में and like उसके बाद 3 दिन का symposium है and कल से शुरू होगा. They provide me this beautiful room and then I'm going to attend the workshop tomorrow morning to learn new things here in this workshop at the Indian Institute of Science. Let's see tomorrow. Okay, bye bye. Indian Institute of Science. Campus is so big.
I can control this. Okay. So let's just try this and see what happens. Maybe I'll make it 100 for example. Okay. So the model is going to take this tokenized input and give output in the token sense only. Okay. So it's going to be again a list of integers. Takes as input a list of integers, gives as output a list of integers. And then I have to now bring it back to human legible strings. Right. So there is this decoder that the tokenizer offers, which will decode from the integer sequence domain into our language. The reverse operation of tokenization. Okay, so let's see what this does. It will take some time. So it's basically put the prompt here. The best way to learn programming is to, this was the prompt. I printed the prompt as well as the response together. And so, this is whatever it is. Okay, it continues on the right. I don't know how to make it sort of word wrap, but it's a long, if you read this, okay, what is this neural network? We know what it's supposed to do. What is it supposed to do? Input is after tokenization. Imagine the tokenizer is separate. Go. Thank you. Split area.
Welcome back everyone for the second lecture of the day. After that brilliant lecture by Aditya, I don't know how I'll follow that, but let me give it a try. So I already gave my introduction in the morning, but just to reiterate, I am from Intel Labs. I'm a computational biology research at Intel Labs, and this lecture is mentioned in the cookbook. Then I have picked a particular recipe to make today. That's like the process of translation or gene expression, where you get that RNA segment. And then once I have made that recipe, whatever is the final preparation, that's like the protein, because that's the real.
Human would probably do with enough time and reasoning, whatever, right? Puzzles and so on. That's sort of the benchmark. In this setting, is it, how would you compare these tasks to what a trained human could do?
Trained humans cannot do that.
Cannot do it.
You can't read hundreds of millions of ACGT and figure things out.
I mean,
right?
given enough tools and so on, and enough time, I think it's not possible.
With tools, maybe it is possible. Like if you develop, I think you'll need maybe several years, but if you develop that domain knowledge of that specific task, and then you have tools to do this, maybe you'll be able to do it.
in such a way that it can create some kind of effect in the cell. So, but the information on these things is very limited. As you talked about generation of some synthetic data, so I want to ask that can some kind of synthetic data can be generated on this to train our model, because the data available for the phytochemical binding to a target is very less. So can we generate some kind of synthetic data to train our LLM model so that it can predict in future that what phytochemical can bind to what target, and some other new targets can be discovered by this, that will open a very new field of drug discovery, that some new lead molecule, some new target can be identified, and that could further be targeted for the drug development and all. So can we use some synthetic data approach here?
So, hello?
Yeah.
First, I just want to mention that I am from a computer science background. So some of the medical jargon or bio jargon I will not understand.
Okay.
So,
so phytochemicals are just compounds, like when we filter out, when we use the particular herbs, we take it through the mass spectroscopy, it produces some chemical compounds. These chemical compounds have some formula, like as we know benzene has C6H6. So each chemical compound can bind to the target based on their side atoms and based on their scaffold. Scaffold means the main core structure of those compounds. For example, benzene is like a hexagon, as we know. So can we generate some kind of synthetic data that can be used for the future thing, that can predict that this that new compound that we discovered can bind to this target and with this kind of efficiency? Can we do that?
so when you say target, do you mean a protein?
Yeah, it's a protein, mainly a protein. It can also bind to DNA as well, depending on the what kind of function it does.
I can give you my take, and I think we should hear from Aditya also.
Yeah.
So, in my mind, if so, so LLMs are also currently being used to learn something like a world model.
Yeah.
Which is that how does the world function, and it could be the physical world, so laws of physics and whatnot, stuff like that, or whatever. The point of that is that if I have a underlying understanding of how things work,
Yeah.
maybe I can generate data which is very realistic,
Okay.
right? So if you have to generate synthetic data, it has to be realistic,
Yeah.
right? Now, if there is enough data already available which covers all kinds of diversity, such that with minor modifications you can create new synthetic data, then I think it would be possible. But if what kind of synthetic data we want to generate, there is nothing similar to that already existing in the data at all,
Yeah.
then I don't know if a machine learning model will be able to generate
Okay.
something like that. As of today, but I don't know. I don't know if Aditya has a different take on it.
I just add something. Yeah, I mean, broadly, I agree with what Sanchit says, in the sense that if you're trying to model, if you're trying to predict within a new context, a new problem setting, you better have some representative data.
Sir,
Now that data can be used either directly to somehow generate new synthetic data. There are ways to do that. There are, for example, models called diffusion models. Even transformers, basically the whole premise is you take some data and learn. They're called generative AI because they can learn the distribution of data. And then you can hope that...
Very sort of statistically experienced, trained people who've been in the field for decades now, who are essentially trying to do this, build these synthetic data sets. So there is a lot of optimism. But again, I share the same doubts. How would the model learn something that's never been seen before? Right? That's not seen. So I don't know the answer to that.
Which a key will fit, assuming it's a very vast molecule. Is my understanding correct? The target object is a very large.
It's flowers like this, and then the target protein which will bind to.
Right. So is the question at which location is it going to bind very properly?
It's a set of protein it will bind to.
Okay. And I suppose the input to this, in some sense, is the structure of the protein.
Exactly.
and the structure of the other object,
Yeah.
right?
Yeah.
Okay.
Yes.
The complexity is because you do not have time to go over all possible.
Mutations that the receptor binding protein will go through.
So this language is a bit alien to me again because I'm not from this, but I'm just imagining like two pieces. So there is a small piece and there is a very large.
Good evening, everyone. And then, like, for first day of the workshop, we just completed the pre-symposium workshop, and like we just learned many things in large language model. Like, it's so good. Bye. Good night.